pleasure to be here and have an opportunity to tell you about some of the work that I'm doing, and along with my colleagues at my institute, on understanding the molecular basis of autism. The Autism Research Program at QBRI is really multidisciplinary. So I'll show you in the next slide the constellation of the excellent faculty we have there. So we're really working on a few different fronts to try and really get a basis of understanding a very complex and heterogeneous disease, nonetheless highly genetic in nature. We have a number of excellent people working together. Let me just put my pointer on here. We start with clinical epidemiology. So one of our faculty is really connected with the clinical community and really doing a clinical diagnosis of the autism cohort families and recruiting them into our cohort, which I'll talk about in a little bit more depth around our human genetics program, again, identifying genes associated with risk for acquiring a complex autism spectrum disorder clinical features. I should mention that we do this in collaboration with the Qatar Genome Program. So the Qatar Genome Program has been really working towards sequencing whole genomes of essentially the entire population of Qatar, around 300,000 people. So I think they're up to 40 or 50,000 genomes now. Healthy genomes, now we're really working towards trying to look at the differences between healthy genomes and disease-associated genomes, which is what I'll talk about around autism spectrum disorders. And then my own work is really around using human embryonic stem cells, pluripotent stem cells, to then create disease models in the dish to try and understand a little bit about the genetics and the molecular biology and the cell biology of the disease, and that's what I'll be talking about. But really, we're a translational organization, so everything we do to understand the molecular basis of disease is really geared towards trying to come up with molecular diagnostics and ultimately therapeutics for addressing autism. So here's, you see the colleagues of mine at the NDRC, Neurological Disorders Research Center, which I'm the director of. So we have people working on neurodegeneration as well as neurodevelopmental disorders. Today I'll focus primarily on our efforts around autism. And a lot of the work that I show you is being done in collaboration with Dr. Young-Soo Park, and I'll say more about his specific work. Much of the upfront work I'll touch on is genetics is really driven by Dr. Kim and his genetics team at QBRI. So the Autism ASD Genetics Project is really a trios-based approach. So we find individuals that present with autism, and we enroll them into our study, and we do whole genome sequences of the parents and the affected child. In some cases, the affected child has a sibling, and we'll recruit that subject as well. So as you know, Qatar is a fairly small country, but nonetheless, we're getting a number of cases, and we're building up a database of the genomes, whole genome sequences of these ASD subjects. We're also starting to branch out and make this more of a regional program. So we've been working with Sultan Qaboos University, recruited a number of subjects from their facility, and we're in late stage discussions, ready to sign an agreement with King Faisal Specialist Hospital to also start recruiting ASD families for these studies. So I'll talk a little bit about the genetics, just really to launch into how we're then doing the functional genomics on the back end. So obviously when you sequence genomes, you find a lot of variants. We're using the parents as the controls in the trio-based approach. So we're finding a lot of inherited, recessive inheritance of genes that we think are good candidates. And I'll talk a little bit about our emphasis on presumed loss of function genes, but obviously with the whole genomes, we're finding de novo variants coming up, copy number variants, and also since we're doing whole genome, we're finding recurrent non-coding mutations as well. So just a little bit about the subject recruitment. Obviously, work isn't possible if we don't have families willing to cooperate and clinical communities that are willing to participate in helping us to enroll this cohort. We're roughly enrolling about 100 families per year in Qatar, 100 families per year in Oman, and a similar number expected to come from our colleagues at Saudi Arabia. And I just sort of listed, within Qatar, some of the facilities that we are interacting with, the Shafala Center primarily and the Ramallah Hospital, who see most of the autism kids that are coming through the system there. And you see here the international collaborators as well. I'll just talk a little bit about autism. I think most of us know a bit about autism. It's becoming exceedingly more prevalent than we originally thought. Probably about more than 1% prevalence. Very heterogeneous clinical presentations. You can see a number of the clinical phenotypes that you see in autism. Every kid is a little bit different. It's a bit of a challenge, to think about it from a molecular genetic standpoint. It's not, one gene causes a disease or even a small number of genes. You read the literature, there's potentially hundreds of genes involved in autism. So anyway, we're collecting our cohort. They're all clinically diagnosed with state-of-the-art standard processes for clinical diagnosis. For this cohort, we exclude those with any kind of major dysmorphic features. We still work with those if they're willing to participate, so we call these the rare neurodevelopmental disorders. But here, I just want to make a few points about the demographics of the cohort. So, we're emphasizing the Arabic population. So obviously, there's a lot of work on autism genetics being done in the U.S. and Europe and elsewhere. And you can see that, with our enrollment, they're pretty much all Arabic, most of them, of course, Qatari nationals. As typical for autism, you see a much higher prevalence in males than females. I mean, one thing here on this point is 26% of our cohort are from consanguineous marriages. So this represents an opportunity when you think about genetics, because we have a lot of recessive inheritance of rare alleles from the population. So it's giving us some insights into some candidate genes that may be missed on other cohorts. And that probably also somewhat explains this prevalence of having an affected sibling, since it's a consanguineous marriage and you have a chance for the other kids in the family. So sequencing the genomes of the other affecteds gives us a better probability that the candidate genes we found in the first proband are also, that genetic mutation is associated with that disease. And you can just see some of the other comorbidities. Not all of them have learning disabilities. Not all of them have problems with speech. One thing we're somewhat interested in is this comorbidity of epilepsy. So I'll talk a little bit about one family where the kids have quite regular epileptic seizures. So this is not just new to us, but I think people have seen this co-occurrence of ADHD and epilepsy with autism. So I'm just going to brush through a lot of work on doing the genetics. We collect the samples, we submit them for whole genome sequencing, we look for, calling the variants, and run through the pipeline, and we're finding, roughly hundreds of variants that are showing up. And this is just, mostly focused on the coding domains. If you look into non-coding, you're talking about even more variants. So there's just a sort of a brief, top of the list, not in any particular order, just to make the case that, we're finding variants. The variants can be de novo, they can be recessive inheritance, they can be point mutations, copy number variants, so on and so forth. And so, when you do this kind of work, you find lots of variants, but then how do you then go and determine that these variants are really associated with additional risk to requiring autism. So that's where the work in my own lab comes in, which is really to try and use human embryonic and pluripotent stem cells as an opportunity to make fully human neurons from individuals that harbor the genetic variations that we've identified by sequencing, or to use CRISPR to add in those mutations into a wild-type background. So I think most of you know about iPS cells, and so you can start with the patient. We usually start with the blood. You take the blood and use the reprogramming factors and turn them into iPS cells, and then those iPS cells can then be directed to differentiate into various cell types. And for the autism work, we drive them to cortical neurons, which I'll tell you about in a minute. But also the opportunity, of course, with gene editing using CRISPR is that, for the iPS lines that we derive and we have a presumptive causative variation, we can go in there and correct that and see if that flips the phenotype, which I'll talk about the phenotyping in a minute. Or conversely, we find this to be a little more practical because often families don't want to be re-enrolled. We identified the gene of interest. We can go in a wild-type background, introduce heterozygous or homozygous variants into those wild-type backgrounds, and end up having an isogenic set of IPS lines, and therefore ultimately an isogenic matched set of neurons in the dish. And so once we have those neurons that we can make at reasonable scale, we can start doing functional and omic interrogations of those. And so I'll talk about how we've been doing that on a particular case study for the purpose of this presentation. One point that I want to raise is how do you select which genes to go after? Like I mentioned, we're seeing potentially hundreds of good candidate variants that are showing up in this, and we're continuing to get more and more sequences and therefore more and more variants. And so you can find a variance more quickly than you can do the functional work. So we can probably in my lab handle about 10 of these, as I'll show you, this functional interrogation per year. So, so we sort of prioritize ones that we think are really novel and important. And what does important mean? It can mean multiple things, but also we want to make sure that it's not something that we'll never see again. In other words, if it shows up in the sibling, then that really strengthens its likelihood. The other thing that's important is what we think the biological plausibility is. Is it a calcium ion channel? Is it a synaptic vesicle protein? And the reason for this is we need to sort of have some working models of hypotheses that are driving how we want to do the functional analysis later. So what I would call experimental tractability. If you're not set up to look at mitochondrial dysfunction, then why go after genes that are involved in mitochondrial dysfunction? The other thing that we keep in mind is what's the opportunity for druggability of these targets. So we don't want to just identify genes. We really are thinking about how we can ultimately come up with treatments for and diagnostics for ASD. So the story I'm going to tell you is essentially from that introduction, how we're taking this for one particular candidate gene, which is called TRPC6. And so this came up as in one of our probands, and it was again homozygously inherited as a recessive mutation from the parents, and there was a gain of a stop codon at the third position. So the presumption is homozygous loss of function of TRPC6. And it wasn't the first time TRPC6 has shown up in ASD genetic cohorts, and so again, there was some reproducibility on this, but we had an interesting proband where it was presumably complete loss of function of this, which I'll show you in a minute is probably true. And the other thing is, again, this sort of biological plausibility and tractability concept is that TRPC6 has been implicated in this stored operated calcium entry. So calcium levels are very important, of course, in all cells, but certainly in neuronal cells for your fundamental activities. TRPC6 works with some other proteins in a complex to help restore the calcium levels, which are mainly stored in the ER. This is an important area of biology, and it's also been previously implicated in other neurological disorders. And my colleague, Dr. Park, who really works on calcium signaling, was quite interested in this. And so, we found the gene, and he's interested in calcium signaling, and so it made good sense for us, and he had the tools to interrogate it. So we went after TRPC6. For this case, we started with a wild type ES cell, and we engineered in that stop codon. So as I mentioned, it was a gain of a stop codon at position 3. And what we found is that using CRISPR, sometimes it's easy to do a knock-in, but often it's more challenging than just knocking out the gene. Since we presumed it was loss of function, we also just did short knockouts. So anyway, we were fortunate in this particular case to get both a knock-in to recreate the gain of the stop, but we also had clones of IPS lines that were specifically deleted for this coding gene. So once we have the IPS lines, then we make neurons from them. So again, these are isogenic pairs. You know, we have the wild type ES line, we differentiate the neurons, and we have the mutated versions that we CRISPR edited, and we drive them to neurons. So here you see just a schematic of the process that we use to make cortical neurons. Again, the presumption is that autistic phenotypes are manifested in the neurons of the cortex. You know, if you're looking at things involved in motor neuron diseases, you would make motor neurons or other cell types. So anyway, for studying ASD, we make cortical neurons, which isn't the most facile differentiation program. You see, it takes us about 3 months to go from an ES starting line. We turn them into neuroprogenitor cells, and then we take about 80 to 90 days to make functional mature, what we call functional mature neurons. And so what I'm showing here is you know, the controls and 2 independent lines, same with the various sets of markers to convince ourselves that we're making neurons. It's not 100% neurons. We have some glial cells in there. We're going to see from some of these stainings. But you know, one thing we need to check on is that, you know, if you have a major defect in something developmental, you may not get neurons from the genes that from the cells that have lost the function of a particular gene. But for TRPC6, we're able to make neurons counting the different types. They're mixtures of neurons counting different types. We don't see any statistically significant difference in the constellation of the subtypes of neurons that we see in there. And then just with this Western here, you can see that in the 2 cell lines that I'm showing you here, there's complete absence of this protein. So indeed, with the knockout or the knock-in of the stop codon, we do have complete loss of TRPC6 in these cells. So now that we can make these cells harboring the mutation with the control, which should be isogenic except for that particular mutation, then we go on and do functional and omic interrogation of those cells. So this is where my colleague, Dr. Park, comes in. So he's an expert in electrophysiology and has a few tools at hand that we bring to bear on not just this project, but other projects. So he can do whole cell patch clamping to look for evoked action potentials in classic means. He also is set up to look at calcium imaging. And we also have multi-electrode arrays where we can look at how cells are connecting with each other. So we take our neurons and we work with, in addition, the lab, and you know, Dr. Park and team will, you know, examine the calcium influx and measure the evoked responses, as well as look at on the MEAs how these cells are networked. So I'm not sure how many people are aware of how the MEAs work, but these are basically wells, tissue culture wells. You plate your cells, and these wells are integrated with electrodes, and so you can probe within the same well multiple independent positions, and so if you get communication between one cell and another, you'll see some synchronous spiking action potentials, and you can also just measure in action potentials in each individual recording. So this is important for, you know, beginning to get some sense of if there's some perturbations in network communication across cells that are plated in these dishes. So let me first talk about, so again, TRPC6 has been, you know, has a role in this SOCE, sort of operated calcium entry. And so the first thing I'll show you is just some data Dr. Park's lab generated with the cells that we've produced. So we make the neurons. One nice thing for us is we can, you know, since we're thinking about development, you know, we can look at earlier, less mature neurons, what we call the 3-week-old, and then at 4, 6, 8 weeks, and interrogate, you know. So some things show up early, some things only show up late, some things are manifested at all stages. So what you're seeing in red are the TRPC knockout cells, and you can see that compared to the wild type, you know, at all time points, differentiation, then you can see that there's a marked reduction in the entry of calcium into these cells. So a clear phenotypic difference between, let's say, wild type neurons and TRPC null neurons. This was another result which we found quite interesting, which is doing patch clamping on, again, the wild type and knockout cells. That, you know, we can look at the evoked action potentials, and you can see here visually with the traces that you see, you know, higher spiking and more frequent spiking in the TRPC knockout, and then we quantify that, looking for no action potential, single action potential or multiple action potentials between the wild type and the knockout cells at 2 different time points of development, differentiation. So not only are they are they impacted with reduced calcium uptake, but they're showing this hyperactivity based on the patch clamping. And then the last electrophysiology experiment that Yang Su's lab did was to we put these cells on multi-electrode arrays, and then you can record the intensity of the spike and whether the spiking is synchronous across the dish, and both metrics parameters being studied. We see that there's hyperactivity of these of the knockout cells. So loss of TRPC is presenting. Peace to you as well. And so we're picking up this phenotype in the dish. So, TRPC6 seems to be important, and these activities, and that's, sort of what we were looking for. But we've also done some transcriptomics, and for sake of time, I'll just go through this quickly. But again, we look for differences between the healthy and wild-type neurons, and we're finding quite a few changes in expression, very significant levels, and you can see, some using gene ontology, you see quite a few numbers of pathways, if you will, that are altered, giving us a concept that we think we're influencing, some sort of, imbalance between GABA and glutamatergic synapse biology. It's sort of our working hypothesis, but this is what the data are speaking to. So what's nice is, when I mentioned about our therapeutic drug screening desires, is that these cells with the knockout of TRPC really become the tool for a screen. So we just actually applied for and received a grant to use this whole setup as a way to screen drugs. So so we have a phenotype in the dish, with the TRPC knockout. Can we use small molecules to then revert that dysfunction, if you will, into a restored normal function? So I just want to wrap up with, I won't go through a whole story on this, but this is another family that we're quite interested in working on. It's a monozygotic triplet family, a Palestinian family living in Qatar. And so we sequenced the genomes of these individuals and found a synaptic vesicle-associated protein, SNAP-B, that has a mutation in the splice donor site. So I won't go too much into this, but we've made IPS lines from both parents and all 3 of the affected individuals. And not only that, but with CRISPR, we've gone into 2 of those affected individuals and reverted that loss of function and so restored that. So here you can see from the Western that the parents express SNAP-B, the kids don't, but if we correct the mutation in 2 of those IPS lines, we restore that function. So now we're doing the functional and the genomic interrogation on these nice sets of isogenic cell lines from that really interesting family, monozygotic triplets. So with that, I'll wrap up. I mean, none of this work would be possible without the generosity of the families that are willing to enroll their kids and their cells in the study. And we work with a number of clinicians to make this possible also because they're diagnosing the kids and then recommend them or, about the study that we're doing, and then we enroll them. Most of the work on making the neurons is done by my postdoc Ali and with a lot of assistance from my RA, Wissal. And as I've been pointing out, Dr. Yang Su's Hart's team is doing all the electrophysiology. So with that, I say peace and open up for questions.